Byzantine Avar relations, Goldenen Stiege, ancient DNA, and haplogroup T1a1a connections across the Caucasus and Levant, Byzantine Avar genetic and historical interactions, 558 to 800 CE, integrating ancient DNA and historical sources. Abstract. This study synthesizes Byzantine, Oriental, and Latin evidence concerning interactions between the empire and the Avar Khaganate, 558 to 800 CE, with new archaeogenetic and biohistorical insights from Austria and the South Caucasus. Analysis of ancient DNA from late Avar cemeteries, combined with reference genomes from the Caucasus and the Levant, supports the coexistence of distinct biological communities within a shared Avar cultural complex. The presence of haplogroup T1a1a-associated individuals in Avar contexts suggests an enduring Near Eastern paternal contributions, echoing Byzantine-Christian lineages and late antique mobility networks extending across the Eurasian steppe. Introduction. The Avar Khaganate's expansion into Central Europe during the late 6th century established lasting Eurasian Byzantine contact zones. Diplomatic exchanges, economic treaties, and conflicts from 558 to 626 CE are richly evidenced in Byzantine historiography. Following the failed Avar Persian siege of Constantinople in 626 CE, the Avar polity fragmented, only sporadic contact persisted, including Byzantine missions in 634/35 and an Avar embassy in 678 CE. Parallel to textual silence, archaeological and genomic data have begun to illuminate the Avar realm's internal diversity and external linkages, materials and methods. Ancient skeletal material from the Goldenen Stiege cemetery, Mödling, Lower Austria, 600 to 800 CE, was analyzed within the histogene's archaeogenomic framework. DNA extraction followed Petrus bone and dentin microsampling protocols. Whole genome libraries were partially UDG treated and sequenced to low coverage less than 1 times. Y chromosomal haplogroups were called on 1240K panels. Mathison et al., 2015. Comparative data comprised the Southern Caucasus transect, 230 individuals, 3,500 BCE to 700 CE, 1516, and the Levantine Iron Age genome from Abel Beth Makkah, sample I2201, T-CTS2860, PCA and QPADM modeling incorporated published prehistoric and historical datasets following eLife's standardized pipeline for Eurasian genomes. Results 1. Byzantine Avar historical continuity. Post-626 textual and archaeological sources suggest Avar retreat north of the Danube and sustained, though limited, Byzantine engagement. Internal strife within the Khaganate led to the Bulgar revolt of 631/32 and fragmentation into Bavarian and Pontic successor groups. 2. Goldenen Stiege Avar population. Ancient DNA reveals extreme genetic heterogeneity in Avar cemeteries. Individuals from Goldenen Stiege show West Eurasian profiles enriched for Southeastern European ancestry, contrasting sharply with contemporaneous East Asian lineages in Lobersdorf graves. One male carried Y-DNA haplogroup T1a1a, a lineage rare less than 0.8% in modern Austrians, but widespread among Near Eastern Christians, indicating limited but persistent Near Eastern input. 3. Southern Caucasus, genetic horizon, genome-wide data from Georgia and Armenia demonstrate continuity with Bronze Age local ancestry, augmented by episodic Anatolian and steppe admixture. Distinct Middle Iron Age males from Kamarakavi, KMR003, KMR004 illustrate gene flow between Caucasian and Near Eastern clusters. 4. Levantine Avar, Y-DNA parallels, phylogenetically aligned with Avar period T1a1a-17. This continuity supports historical models of Byzantine, Levantine, male mobility, and conversion contacts linking Eastern Christianity and Avar frontier elites. Discussion: the Avar Khaganate embodied an empire without genetic uniformity, a cultural amalgam binding East Asian steppe and Mediterranean ancestries. The T1a1a male of Goldenen Stiege occupies a liminal genetic niche between the Southern Caucasus and Near Eastern Christians, paralleling the hypothesized Byzantine mediating channels of recruitment or alliance. Such results underscore the limitations of purely archaeological definitions of Avar and reinforce genomic evidence for a plural, multi-ethnic polity consistent with historical testimony of subjugated Slavs and Bulgars. Comparative analyses of Caucasian data further reveal that trans-Caspian links persisted well into the migration period, while Levantine-derived haplogroups such as T continued diffusing through imperial, religious, and mercantile networks. Conclusion: the convergence of historical and genomic evidence reframes the Avar Khaganate as both a successor and partner to late antique Eurasian mobility. Shared paternal markers such as T1a1a, attested in Avar, Austria, and Levantine Caucasian contexts trace enduring genetic footprints of Byzantine-linked Near Eastern lineages at the fringes of the early medieval world.